Hello, welcome to this second Albrecht Müller ARRIGE seminar. This year, we will devote this ARRIGE seminar in honor of one of the founders of our association to applications of the developments of genome editing of animals, livestock, and pets, xenotransplantation, and improvement of animal performance. Unfortunately, due to an overlapping commitment, I will not be able to be with you, but I'm sure you will enjoy a fantastic seminar with the help of the rest of the ARRIGE board members and all the collection of presenters and invited speakers that have accepted to share their knowledge. Therefore, please enjoy this second Albrecht Müller ARRIGE seminar. Bye-bye.
Okay, ladies and gentlemen, we are going to start. Thank you for joining us for this second Albrecht Müller seminar. You all know our association, ARRIGE, which is working for the development of a responsible research and a responsible development of genome editing in the various fields. And today we are going to dedicate this afternoon to some recent aspect, innovative aspect of genome editing in the field of animals. I'd like first to thank the organizers of this day. I am speaking as the vice chair of the association. Luis Montolio is requested on another meeting and couldn't make this introduction, but I'd like to thank particularly François Hirsch, because he was the one to initiate this Albrecht Müller seminar, and is really the one working very hard with the people of the board. I see Bongi, I see Julian. You don't see Christine Lemaitre, but they all are working hard, and they were working hard to contact all of you to make this seminar possible. So thank you very much for their dedication and their work. And also, just to remember that this seminar is in the memory of our colleague Albrecht Müller, who was at the very beginning of ARRIGE. At the very beginning, some of you were also involved. We were traveling all around the world, particularly in Latin America. Martina, you remember, particularly in Africa. We made a seminar in Brazzaville, particularly in India. And Albrecht participated in some of them, and sadly he died a few years later from glioblastoma. So in his memory, and because we need to continue this responsible research and innovation, thank you all for joining this afternoon. And without any more delay, I let the floor to the first session to start the real interesting meeting. Thank you for participating. Thank you for your questions. Please ask the questions through the various ways you have on your line in the bottom of your screen. And let's go for session one.
Thank you for the introduction to the conference today. My name is Bruce Whitelaw. I'm the director of the Roslin Institute in Edinburgh, UK, and I've been working with genetically modified animals for all my academic career, and latterly this is focused on the use of genome editors. I am passionate about these tools. I'm passionate about what it can offer to our society through my research through livestock, and from the speakers today, I think you'll gather that there are many exciting applications and there's much passion around it. Anyway, going back to the idea of responsible dialogue, this is what today is about. We have 4 speakers in 2 sessions. I'm going to introduce The first two speakers and my good colleague Martina Crispo will introduce the second two before we have a round table session. But before we start that, I just want to set the scene. And that is, it's a really exciting time for genome-editing livestock. It's caught the attention of politicians, it's caught the attention of the public groups, it's caught the attention of the media, and it certainly caught the attention of science. We have within both Europe and the UK ongoing assessment of whether the current regulations around GMOs are fit for purpose for genome editing applications. Other countries around the world are ahead, if you want to look at it that way, and have already put in place some regulatory processes. Indeed, there are even products available in some parts of the world. This is the time to have the dialogue. This is the time to reach out and communicate and to learn what are the hopes, what are the aspirations, what are the concerns, and how we can come to a responsible position of conveying all that. The speakers come from, well, I know them all, but that's by the by. They come from two countries, and that does reflect to a certain degree the amount of effort that these two countries, Scotland and Germany in this case, apply in this field. But there are many other actors around the world. And the topics you'll hear will certainly give you the breadth of what's going on within livestock, from reproductive technologies, disease resistance against virus infection, through to models of human disease and xenotransplantation. It's not exhaustive of what's happening, but it's going to set the scene. What I hope for is a very fruitful discussion later on in the afternoon. We will have a fairly tight schedule, and it's Martina and my task to try and keep the speakers to that. Each speaker will, and the first four speakers, each speaker will have 15 minutes to tell us about their wonderful research, and then 5 minutes for questions. Now, that 5 minutes may disappear very quickly. Do not worry, there's later opportunity, more opportunity later on in the day where we can bring these to the fore. I would also say that it's not, although the first four speakers come from two countries, it's not a geographically restricted topic. This is a worldwide topic where regulations that happen and activities that happen in one country impact on what happens elsewhere. And we have to be aware of that. Ideally, we want to get to a stage where this harmonization across regulations that has proven difficult for most things in our world. However, we get a certain degree of harmonization, and I think that must be something we aspire to. It's very active in the United Kingdom just now. For those of you who've been following the bill, the precision bred genetic technologies bill was in the House of Commons yesterday and is currently in the House of Lords, and we are waiting to hear the outcome of that. There is much activity around the point from all communities, and I think that is a very good, fruitful, and productive position to be in. Right, I will stop here and we'll steal a couple of minutes on the agenda, and I'm going to ask Angelika Schnieke to introduce herself first, and then tell us about her science. Angelika has been working in this field since her PhD, to be honest, if not slightly before. Angelika and I have crossed and interacted over many times. One of the world leaders in genetic engineering, large animals in particular, and pigs, and I leave it to you, Angelika.
Okay, I wasn't quite sure if Björn was going first. Sorry, is that no change?
My apologies. Björn, you want to go first, by all means. I think I was showing my chivalry there rather than my reading of the rules. My apologies. Björn, please come first. So I'll give away a little secret. Björn is, I'm not going to say in detail, but Björn is just about to change job. Congratulations, Björn.
Thank you. Thank you so much. So yeah, I'm Björn Peterson. I'm working at the Friedrich Loeffler Institute in Mariensee. the Friedrich-Lafley Institute is the Federal Research Institute for Animal Health in Germany, and I'm working at the Institute of Farm Animal Genetics, and I've been working in the field of genome editing of livestock species now for almost 20 years. And I'm thankful for the invitation to give a talk here today at this ARRIGE webinar. And I will talk about the advances and current barriers in xenotransplantation. So allotransplantation has been a great success since the first heart transplantation in 1967. And the problem is that despite several efforts to increase the number of available life-saving organs, there's still a huge gap between people on the waiting list waiting for a suitable organ and transplantable organs. And that leads to the fact that each day more than 3 patients die in the EU while they are waiting for a life-saving organ. And people on the waiting list, after 3 years, almost 17% of the patients have died. So there's an urgent need for other options to overcome this problem. And there are 3 technologies discussed so far. The first one is the tissue engineering using ES cells or iPS cells to generate organs in vitro and then transplant them into patients, which is still at a very preliminary stage. So far, organoids can be produced by this technology, but it's still a far way to go to produce a transplantable organ. Another option that is discussed is bioprinting, to use cells to print an organ. It's also at a very preliminary stage, but it has some nice features that can bring it or can be a solution for the future. And something that is close to the clinics, I would say, and I hope to convince you that it's really close to the clinics, is xenotransplantation. Many of you may think that xenotransplantation is something completely new, but it's even older than allotransplantation. So the first recorded xenotransplantation took place in the 17th century when they used bones of a dog to treat an aristocrat's skull fracture. And even in the last century, several xenotransplantations were performed where they used organs from non-human primates and from primates to transplant the organs into human patients. And there was quite a good survival time. So one patient survived for 9 months after receiving a chimpanzee kidney. And the last xenotransplantation was performed in 1984 when baby Faye received a baboon heart and survived for 20 days and then died of multiple organ failure. So as you can clearly see, xenotransplantation is not something completely new, but it was facing severe problems because with non-human primates or primates, you have ethical concerns. You do not have the technologies to edit the genome of these species. There's no real reliable cloning technology for these species available, and you also run into problems regarding zoonosis spreading and so on. And you cannot easily propagate these animals and keep them under very highly hygienic conditions. So scientists were thinking about alternatives to using non-human primates and considered the pig to be the best organ donor for xenotransplantation. And that is the case for its high similarity regarding genome, the physiology, and the anatomy to humans. It can be kept under highly hygienic conditions. The organ size is quite similar to humans. There are less ethical concerns as we use these animals for thousands of years for meat production. They have a short generation interval, a rapid growth, and also the transmission of pathogens is considered to be of minimal risk. The real problem when you think of transplanting a pig organ is that severe rejection responses occur. And the reason for that is that the pig has several epitopes on their cell surface that cause these severe rejection responses. These are carbohydrates, sugar molecules that are expressed on the cells of pigs, but are not expressed on human cells anymore. We lost the genes that are responsible for these gal sugar during evolution. So about 1 to 3% of all circulating preformed antibodies in the human blood flow are directed against, for example, gal sugars. And they bind to these sugars after transplanting a pig organ and activate the so-called complement cascade, which leads to the destruction of the transplant within a few minutes. So during research in that area, several other antigens were found, like NEO5GC and also SDA, which also trigger rejection responses. And beside these epitopes that are expressed on pig cells, we are also having some incompatibilities between the human and the porcine, for example, coagulation system. These are important factors when you can overcome the hyperacute rejection response. That is then the acute vascular rejection, but it can be addressed by expression of protective human genes, for example, human thrombomodeling, hemoxygenase 1, EPCR, or A20. So the last rejection response that would occur is a so-called delayed xenograft rejection, which is triggered or caused by the acquired immune system and causes anti-pig antibodies and cytotoxic T cells that is quite comparable to the delayed rejection response that also occurs after allotransplantation and can be overcome by a good immunosuppression, but can be also addressed by expression of transgenes that lower the responsiveness of T cells against the xenotransplant. So as you can see here, a lot of genetic modifications are necessary to make a pig organ suitable for xenotransplantation. And that was a great barrier for decades, but now we have new technologies in our hands, as Bruce already mentioned. We now have genome editing tools like the CRISPR-Cas system in our hands, with which you can easily address every locus in the genome, and you can modify it by knocking out genes, for example, the genes that are responsible for the expression of these antigens on pig cells, or you can introduce human transgenes at a specific locus in the human genome, in the pig genome. And another important technology is the cloning technology, which was first described with Dolly the sheep, which is necessary to perform targeted genetic modifications in the porcine genome. With that technology, we can use somatic cells from pig, do the genetic modifications in vitro, test the cells before we use them to produce a living offspring, and then use these cells for somatic cell nuclear transfer to produce a pig that might be suitable for xenotransplantation. So these technologies have led to 10-fold or 11-fold genetically modified pigs that are now available for xenotransplantation studies. This is just an example from one of the companies that are trying to bring xenotransplantation into the clinic. And they have produced pigs that have a full knockout or 3 knockouts, then a perf knockout, which has about 15 to 25 integration sites in the porcine genome, and it expresses 9 transgenes. So this shows you how powerful the technologies are that we have in our hands now. So after producing such a edited pig, you first test the cells from these pigs in so-called in vitro assays, like a complement mediated lysis assay. Here you can see that these edited cells are much better protected against a complement mediated attack. And we also test organs of these pigs in ex vivo perfusion systems where these organs are perfused with human blood, and you can measure several parameters that are indicators for rejection responses. And after these experiments are successful, you are using organs from these pigs for a pig to non-human primate xenotransplantation, and where you study the rejection responses after transplantation of these organs into non-human primates. And then the last step would be to perform clinical trials. But there are some recommendations from the FDA, for example, where you have to achieve a survival of longer than 6 months in 6 out of 10 consecutive life supporting trials before you can apply for a clinical trial. So beside the genome editing, there are also other factors that can influence the outcome of a xenotransplantation, and that is the preservation of the organ that has to be transplanted, and also to develop new immunosuppression regimes that are suitable for xenotransplantation. There are several immunosuppressive drugs now that are tested in research, but still have to be tested in clinical trials, like the anti-CD40 antibody. But it has been shown that these immunosuppressive drugs are a great advantage for xenotransplantation. So how far are we so far? So this is data from pig to non-human primate transplantations of different organs like kidney and heart. Heterotopic means that the heart was transplanted. It was not a life-supporting transplantation. It was transplanted into the abdomen, so the baboon still kept its own heart, and the pig heart was transplanted into the abdomen. So there, a survival time of 945 days was achieved. And when you remove the baboon heart, and replace it with a pig heart, survival times of 195 days and 264 days were published so far. So these experiments fulfill the requirements from the FDA to go the next step into clinical trials. So at least 10 years ago, scientists said that clinical trials for xenotransplantation are anticipated within the next 5 years. So this is still what I would state now, but you never know. So the first xenotransplantations have been performed. So a pig to human xenotransplantation of a kidney has been transplanted to brain dead patients and have been perfused for 54 hours and 72 hours without any signs of rejection so far. And if you might have all heard, this year, the first successful pig to human xenotransplantation was performed in the US, where a 10 gene edited pig heart was transplanted to a 57-year-old patient named David Bennett, who died 2 months after transplantation. The histologic examination of the heart revealed a scattered myocyte necrosis, interstitial edema, and red cell extravasation. The patient was not eligible for a allotransplantation because he didn't obey all the recommendations from the doctors for examinations and taking his drugs and all different things. He was on ECMO for I think for almost 2 months. So he was in a very bad body shape, and this was his last chance to survive. So he admitted to receive the pig heart.
1 minute, Bjorn.
Yeah. So one problem that was obvious during this experiments was that the pig heart was infected by a porcine virus, which is CMV, and might have caused some problems. So they couldn't address this problem with several drugs. And there was something that we also observed in non-human primate studies. When CMV-infected pig hearts were transplanted, the survival rates were much lower. So CMV is something critical and should be avoided. There are test regimes available to avoid transplanting ZMV-infected pig organs. So xenotransplantation is just around the corner. The corner might have been much shorter now. There's several companies now around the world that are spending a lot of money to bring xenotransplantation into the clinics. Here are just a few displayed. And yeah, to sum up, we have now very sophisticated technologies in our hands to edit the porcine genome to produce pigs that might be suitable for xenotransplantation. We have advanced immunosuppressive regimes to further reduce the rejection responses that might occur. So there are still further improvements needed for the matching of patients suitable for a xenograft and the approval of BMP requirements. But I'm sure that we will see xenotransplantation soon into clinical trials. And for that, I would acknowledge all my colleagues here in the Institute and our partners in the German DFG-funded xenotransplantation project. And thank you for your attention, and would be happy to answer your questions. Thank you.
Thank you, Bjorn. This is the moment we all clap you, so thank you very much for that. Please, if anyone has questions, please post them in the Q&A through the facility, which you'll find at the bottom of your screen. But maybe I could just ask you one question to start off with. So the goal here is to increase the number of organs, where it's still going to see immunocompromised patients or the patients that are given transplants being immunocompromised. It's probably too long an ask, or too big an ask, to completely remove the need for immunocompromisation. So that's the first question. Do you see it always being delivered to a patient that is immunocompromised? The second question relates specifically to genome editing. This tool that we have, has it sped up the process? Has it allowed progress to be made quicker? Or is it just, if it's not doing that, or indeed if it's doing that as well, is it allowing more applications to come to market? What is the impact of genome editing? But first, do you see the use, the application is really to increase the number of potential organs for transplant, rather than the aspiration of removing immunocompromisation as part of the treatment?
I mean, the question is if you really can reduce the immunosuppression. I mean, as you have seen, we use immunosuppressive regimes right away, but maybe you can reduce the number of immunosuppressive drugs that have been taken by the patient, because what you can do also is to better monitor the pig before you transplant the organ. You definitely know what kind of pathogens are in the pig. You can keep them on a very highly hygienic conditions before you transplant the organ. That is not, that cannot be done for an L transplant, for example. I mean, they can just make some quick checks of some severe pathogens, but you cannot test for all the spectrum, I would say. The impact of the genome editing, I think it has, for sure, it gives us completely new opportunities now. Before we had the genome editing tools, it was really hard even to achieve a knockout. I mean, with just homologous recombination, it was like finding the needle in the haystack to find the right cell to be used for producing a knockout pig. And it was not really achievable to have a targeted integration of large cassettes of transgenes at a specific site in the pig genome. So we can address these problems now with the genome editing tools, and we can do multiple genetic modifications at one shot. So that was also not feasible. So like the perf knockout, for example, you can easily knock out 15 to 25 sites in the genome. That was not feasible before the genome editing tools. So it is now, it's much safer. We can now target what we want to target, and that's it has a great impact on xenotransplantation and the production of genome edited.
Thank you, Bjorn. We have a question in the Q&A box, and the question is: are there organs that are more likely to generate a rejection response than others?
I mean, for sure. I mean, there's some for example, islet cells or something like that. I mean, you can transplant islet cells, for example, at a side of the body that is protected against the protected from the immune system, or you can encapsulate these organs, for example. I mean, likely to organs likely for rejection responses are organs that are connected to the blood flow, because then they have direct contact to the antibodies, to the preformed antibodies that are in the blood flow. So there are some that are more likely, and there are some other organs that are more likely to produce problems regarding the incompatibility between the different systems, for example, like the coagulation system.
Thank you very much. Björn, thank you for your presentation there and kicking off this symposium today. We'll move on now to Angelika Schnicke, and Angelika will maintain the theme of using genome editing tools to create animals for biomedical applications. Angelika, if you could do a quick introduction to yourself and then give us your presentation.
So I hope you can see my presentation and you can hear me also.
Yes, and yes.
Yes, good. So my name is Angelika Schnicke. I have been, like Bruce probably also said, working with genome modifications all my life. And this is now quite a long time ago that I started first working with mice, then at some point with cattle, with turkeys, and now in the end also working mainly with pigs. I'm afraid you already heard the talk from Björn, and so there will be somewhat repetition now. Principally, Björn has already said most of the important things when it comes to xenotransplantation. So organ transplantation, as you can see on the slide here, is the solution for end-stage organ failure. And just to repeat what Björn had said, we can do organ transplantation now relatively efficiently. You can see it here. The survival for organs, the heart, 5-year survival is close to 80% for some organs, like here the kidneys, we're already over 80% of the patients. One patient who dies and who can also present it, their organs can be used for organ transplantation. One patient or one person can then really help 8 different patients. So you have 1 heart transplantation, you can give 2 kidneys, 2 times the lung, and so on. And still we have a problem, as Björn said, that we have not enough organs. And you can see it here. This is for Europe. You will have much bigger number if you go worldwide or if you go to America. So what can we do? We can, of course, donate our organs. And you can see on the slide here that some countries, like for example, Spain, is very happy to donate their organs. Other countries like Germany is not so efficient, it's less than 10%, and during Corona, these numbers were still going down. And even if everybody would be able to sort of, at the end of their lifetime, say, I would like to give my organ for organ donations, we have one problem, that most of us will die of old age, and so only maybe 3 people out of 1,000 who die can give their organs for organ donations. So we need an alternative, and one of the alternatives, as Björn already mentioned, would be the pig. And we will now talk about what we need to do to humanize the pig, something about the safety towards the clinic, and a couple of alternatives. Most of it you have already heard. So if you come to normal transplantation, we mainly have the problem with cellular rejections, and this is mainly due to activation of the T cells. Xenotransplantation, we have hyperacute rejection, which we heard is due to preformed antibodies. Some of these are called the alpha-gal antibodies. What can we do? We can inactivate them. In addition, we can lower the complement activation by adding genes like complement regulator genes, which then would again suppress the rejections. We come then to the acute humoral rejections, which doesn't happen in days, but in weeks. And again, here we have non-gal epitopes, which we could eliminate, and also then problems with discrepancy between the pigs and the humans. So where we have the incompatibility, we might have to add human genes. And then finally, we come to the cellular rejection. So you can see we have to do multiple knockouts and add also multiple genes. And for me, this is sort of summarized in the slide here. I have started with xenotransplantation when I was first in Scotland, and this is now many years ago. And sort of about that time, 35 years ago, all we could do is DNA microinjection. And that meant we could randomly integrate some genes into the genome of pigs. And whoever has worked with pigs knows that DNA microinjection, because the pig embryo is black, was incredibly inefficient. Less than 1% of the embryos which went to birth was indeed transgenic. So all we could do, maybe add a couple of complement regulator genes. That meant we sort of could suppress the hyperacute rejection. We could do nothing about endogenous retroviruses, and we could not place any transgenes. If we then go back sort of 25 years ago, when the nuclear transfer was established, we now could have transgene edits a little bit more efficiently, and we could also do the first knockouts. And the first knockouts ever done in a pig was indeed then the alpha-gal knockout. So with that, we could come over the hyperacute rejections. We still had the perfs, and placing transgenes was very inefficient. And then the genome editing came, and now we can inactivate simultaneously multiple transgenes. We can add multiple transgenes, integrate them at the right place. We can do this in a homozygous way. And so we can overcome the hyperacute rejections. We can also eliminate the perfs and also do the transgene placement. And I thought for this group, it might also actually be important to sort of realize what does it mean when you work with large animals. So here we have now done in the old-fashioned way one of the typical knockouts. That means that most of the knockouts, of course, were only heterozygous. We then had to start breeding. So the next generation, we again had only heterozygous animals. We then had to breed again to get to the homozygous animal, wait until that animal has sort of maybe 5-6 months of age, and then we could possibly use the organs. And you can see it takes 2.5 years. But then we don't only want one knockout, we want also the second knockout. So again, we can do the same thing. We can breed the animals another 2.5 years, or we can take one of our already generated knockout animals, breed with these animals. And you can see it already starts getting very colorful. This means we have to breed a lot of animals so that we get maybe within 5 years to a double homozygous knockout. Yeah, but we don't only want the double homozygous knockout. We want to have maybe triple knockouts, we want quadruple knockouts, and in addition, we want also then transgenes. So the breeding in the end got so complicated that some people could only generate the animals again by nuclear transfer, because otherwise you had too many animals. So once you accept that xenotransplantation is something you want to do, then using now CRISPR-Cas really makes it a lot more efficient. And from an animal point of view, the number of animals used for experiments has really been reduced. And I will show you one of our experiments and also of other people where you just do in one single step a quadruple knockout. And this is then a homozygous knockout. So in one single generation, you have a 4 times homozygous knockout. So how did we improve xenotransplantation? You can see here that was work from Konrad in our group. He made the first animals where we actually combined a number of transgenes so that all transgene was in a single locus. You can see it here. It was the complement regulators, CD46, 55, 59, hemoxygenase, and A20, sort of to stop activation of the endothelium. If you look at the effectiveness of the transgenes, down here you can see that once you add human serum, these animals were almost protected from lysis. In the next step, we also thought, can we now also put transgene at a particular locus? We did transgene placement at the ROSA 26 locus in the pigs, and we could, with the old methods, target the ROSA 26 locus. And again, if we put our complement regulators into this locus, they protected the tissues from serum, from complement lysis, sorry. Well, Targeting to a particular locus, as I mentioned, we can do it the old-fashioned way. We have about 2% if you go into the Rosa 26 locus, which is very efficient to target. But then we also tested targeting the same locus using now the CRISPR-Cas for gene placement. And you can see our efficiency now to place our genes at that locus is more than 30% of the cell clones. Then Konrad did another experiment where he indeed wanted to knock out 4 genes. And this was one of the animals which was born with 4 homozygous knockouts. And actually, I was corrected when I gave this talk once before that I hadn't inactivated 4 genes. We had actually inactivated more genes. So if you want to inactivate the B2M, there might be pseudogenes, which also had been inactivated. We also did a different type of inactivation for the SLA1 locus, where we had 2 more pseudogenes, and all of them were inactivated in one single step, and they were homozygously inactivated. And you can see here again, wild-type animals, cells from wild-type animals. If you added human serum to them, you can see that complement, or in this case also antibodies, were deposited on the cells. If you had 2 knockouts, fewer antibodies, and if you had all 4 knockouts, you had actually relatively efficient clearance of the antibodies. So I mentioned that it was about 5 years ago that really genome editing took place in the large animals, and also especially for xenotransplantation. And these are the publications which came out since 2017. And what you can see here is that people now could do 3-fold knockout in one go, 4-fold knockout in one go, and it's the same time placement of genes. And this was, for example, down here, which also was mentioned by Björn, you can have a 3 times knockout and at the same time place 8 transgenes in one of those knockouts. And in the last one, you had the 3 times knockouts plus 9 transgenes, plus then the knockout of the perfs. I added this slide briefly because Bruce also mentioned that can you actually make the organ so that you don't need immune suppression? And one advantage is for the xenotransplantation that you can actually have gene expression, not only complement regulators, but you could also get medications, for example, recombinant antibodies or the CTLA4 Ig, which can inhibit the activation of the T cells directly expressed, for example, in islet cells. So they now express their own immune suppression. You can then do the transplantation and you don't have to give general immune suppression anymore. In our group, we're also working on something which we call smart graft. We have tested it in mice. It seems to be working, where you can have an inducible promoter, which you can see down here, and it gets activated by cytokine release. So if the organ now starts a rejection, produces cytokines, or the human against the organ produces then cytokines, the promoter gets activated, and you make your own immune suppressant drugs. And so this can be local, it doesn't have to be systemic anymore. So then we come to the safety, and here again, one of the original worries was that we have endogenous retroviruses, both the humans and also the pigs, and the pigs it's then called perfs. We have found that animals, there are some animals which are free of perf-C. This is the one which is sometimes still active. A Chinese group has shown that you can inactivate, not eliminate, but inactivate 25 perfs in one go. And this again is CRISPR-Cas. Or if you have animals with small numbers of active perfs, you could actually eliminate those copies. And when it comes to healthy animals, we of course have to also ensure that they don't have any infectious diseases which can go and infect humans. And one of those is, for example, the cytomegalovirus. But pigs you can keep in SPF facilities or also in DPF facilities. You could even have them in gonotobiotic facilities, but that's not so good if you want to keep a herd bird. Towards the clinic, I don't have to say very much, because Björn had already talked about this. We have here, for example, the bovine heart valves. They're already in the clinic, and also porcine heart valves. The islets have been tested in the preclinical trials and also in humans already, but these you can encapsulate. Preclinical trials have happened in baboons. And one important thing which we shouldn't forget when we talk about xenotransplantation is that you don't have to have the organ for the rest of your life. It could be a bridging organ until you have a human organ, or in some cases, there are also suggestions that it could be an ex vivo perfusion, so that, for example, you have a liver problem, your liver can regenerate, it might need time to regenerate, so for that time, you're hooked up to a porcine liver to take over the function.
1 minute.
Yep, I'm almost at the end. This one I can skip because Björn had already showed how long the organs survive in the pigs. We had then the pig organs survive in the monkeys. We then had the first brain-dead organ transplant into humans. Björn has mentioned that. This here shows which organ type it was. And again, you had here 4 knockouts, all done by CRISPR-Cas. And then we had the first transplant into humans. Again, Björn has already mentioned it. We have alternative technologies, which were also mentioned by Björn. And maybe I also should mention that also for other projects, we can use it very well. Here, for example, we're working also on cancer. We wanted to have Cre expressed from particular endogenous genes, and we could use then CRISPR-Cas to place our Cre genes into the locus of, for example, the PTF1 gene to make animal models for pancreatic cancer. We have also produced animals which have the CRISPR-Cas located now in the ROSA26 locus. We can then show that this is functional, and now you can do by just adding the guides. You can do in vivo genome editing. Here, for example, we eliminated about 12.7 kb from a gene which is important for heart disease. And we could show that you can do that just by adding AAV to the heart. We have to still work on the efficiency of the vectors. We can use it to produce animals. These are 3 of the PhD students who really did the whole project from beginning to the end, from injecting CRISPR-Cas into the porcine oocytes, which you can see here. You then had immediately heterozygous and homozygous animals which were born. In this case, it was a model for IBD, where we just deleted a small fragment. And you can see here the disease, which we had then, the IBD, was very similar to what you find in humans. And you can also do analysis of the microbiome. So with this, I will stop now. You can see that large animals are actually not used that often in research at all. It's about 1%. But of course, they have a lot of advantages. We can use them for organ transplantation, for surgery, where they have similar kinetics, pharmacokinetics to humans. The immune system is very similar. We have already various disease models. They're ideal for cardiovascular disease and also for nutritional analysis. So there's sort of a bridging from the basic science as a translational models towards the clinic. The importance is sometimes also the size, not always, but whenever it comes to instrumentation, to imaging, biodistribution, longitudinal studies, and of course, organ transplantation, the size does matter. And we could also give you quite a number of samples that there are species differences, that sometimes the pig models is closer to what you have in the human disease than you would have in the mouse. And with that, thank you very much for listening.
Thank you, Angelica. And again, the virtual round of applause for you. Please type any questions you have into the question and answer, but I have, it's more a clarification, which I know the answer to, but I'll just see if you can, at the end of Björn's presentation, he showed some of the companies which are taking this technology forward. The research to date has been funded primarily by public funds. Could you just give some short commentary on how the research has been funded to date?
I think it's slightly different how it is in the different countries. So in Germany and also in Europe, it was mainly either EU funding or local funding. So here in Germany, we have the DFG, where both Björn and I are in a large consortium. In America, there was quite a bit of funding also from industry, a lot more than here in Europe. So he mentioned eGenesis. eGenesis got large amounts of money from, I think, Fresenius. There is what used to be PPL Therapeutics in the States, which is now Revivicor, and Revivicor has also mainly funding from industry. And these are also the animals which had gone now into the first human trials, both into the brain dead patient and also into the human life patient for the heart transplant.
I'll sneak another one in. Both presentations were about pigs. Could you give a comment about the likelihood of any other farm animal species being used, or is it simply that the organ size of the cow is too big, the sheep is too small?
Depends sometimes on the organ, of course, you're working with. The whole gastrointestinal tract would not be very suitable if you go for either the sheep or the cow. Cows are, of course, too big, and also the generation time. You have in pigs, you have a litter, while in the other animals, you only have 1 or 2 offspring. And an important point is also the pigs you can keep in SPF facilities, which you cannot do with either sheep or cattle.
Thank you very much. I realize my time is up and I'm going to have to pass on to Martina. But before I do, I just want to thank both Bjorn Pedersen and Angelika Snicker for a very illuminating talk, covering a number of aspects of this technology and showing us clearly that xenotransplantation, although it's had quite a journey, is actually here and on the ground and is happening. So thank you both to you, and I'll give you a wee clap. Martina, I'll pass over to you now.
Thank you, Bruce, and thank you for the nice presentations. And now we are moving to the next roundtable of why using genome editing for improving livestock performance. So our first speaker will be John Otley. Hi, John. Now I see you. And I will make a brief introduction for you. And John is Associate Dean for Research at the College of Veterinary Medicine and Tenured Professor in the School of Molecular Bioscience at Washington State University. His research program focuses on understanding the biology of germ cell development and genome engineering of animals. And he is also a recognized voice for the application of biotechnology in food animals. His talk will be about genome editing reproductive capacity in livestock. So I leave the floor to you. You have 15 minutes and 5 minutes questions. So I will tell you when you have 1 minute left. So thank you, John.
Okay, very good. Thanks for the introduction. I'll share my screen. Okay. Just to make sure you can see my screen, everything's fine? Thumbs up?
Yes, yes.
Okay.
We see.
Perfect. Okay, so just again, thank you for the opportunity to share some of the perspectives that I have on how gene editing can be applied to livestock, and I'm happy to have any discussion afterwards. So I want to start by defining how an animal scientist like myself views genetic engineering of animals, and that is the direct manipulation of an organism's genome. And this could be sophisticated through tinkering and altering DNA directly, or it can be conventional through reproduction, in which the genome that is carried by a sperm that has one set of chromosomes is combined with the genome carried by an egg to now make a diploid organism that can go on and sustain life. And through either the use of gene editing or selective breeding, human intervention is needed to engineer the genome of an animal. And so we can trace the origins or the dawn of animal genetic engineering really to the domestication of animals. And this was first started with the dog some 15 to 30,000 years ago, when humans started to intervene in the breeding of these animals. And what followed was then the domestication of livestock or food animals, including sheep, goats, pigs, and cattle. And this is somewhere between 8 to 10,000 years ago. And so we can really see the origins of human intervention into animal breeding And that would be genetic engineering, bringing together genomes that may not have created in nature because of human intervention. And this can be dated all the way back to ancient civilizations, such as this cave painting from ancient Egypt, dating back to almost 12,000 years ago, that indicates that humans were intervening in how animals were being raised and how animals were being bred. And so in more modern times, we can observe the impacts of selective breeding on livestock by simply appreciating the changes in phenotypes over relatively short periods of time. And we can see this in cattle breeding. And so this drawing depicts the Grand Champion Shorthorn breed of cattle in the United States in 1835 and then in 1937. The big animal is in 1835 and the smaller animals in 1937. So in roughly 100 years of selective breeding, the frame size was dramatically changed by human intervention. And in even more modern times, we can see in short periods of 30 to 40 years, how breeding, selective breeding by humans and cattle can dramatically, again, change their phenotype. So between the 1950s and the 1980s, just 30 years, these are all Angus cattle. In just 30 years, changed the frame size dramatically through selective breeding. And then started to change it again over the next 30 to 40 years. And these are all Angus cattle again. So you can see how the frame size was shrunk and then giantized again, and then started to shrink again, and a sway was put into the spine, all because of human intervention in the breeding of these animals. And so the reason that we do this is to try to address food security and really trying to stave off insecurity and solve problems of insecurity for feeding the human population. And so even though greater than 2 billion people on the planet are classified as overweight, roughly 90% of those people live inside high-income countries. There's also roughly 1 billion people that are considered malnourished across the planet, and almost all of them, 98%, live outside high-income countries, so in middle and low-income countries. In fact, when you calculate the number of deaths worldwide based on starvation, cancer, malaria, foodborne illness, or HIV/AIDS, the number of deaths by starvation is more than all of those other problems combined. And so this really is a global food challenge. It's a global problem on how do we address food insecurity. And it's not just a problem now, but it has a large looming issue as well, and that is through population growth. So this is data for the billions of people on the planet between 1900 and a prediction out to 2050 based on historic growth trends. So somewhere around 1940, the world's population began to double. So between 1940 and 1980, it doubled. And then 40 years later, 1980 to 2020, it doubled again. And we're predicted still to hit over 10 billion people by the year 2050. Well, to feed 10 billion people by 2050, there will be a need to increase food production by 60%. And this is both crop and animal protein. And so a solution is not simply just using more land, because these data show that as population grows, the amount of land that's available to increase agricultural output, whether it's cropland or grazing land for livestock, declines. And so we can't simply just open more land or throw more animals on land. And so the solution is going to be multifaceted to address this global problem of decreasing agricultural land, but increasing a demand for food. And the demand for animal protein continues to increase. Between 1970 and the year 2000, the demand for animal protein rose by 70%. It rose by another 50% over the next 20 years, and it's continued to grow out to 2050 as we work towards 10 billion people on the planet. And this is especially true in low-income countries that are moving into mid-income status, and mid-income countries that move into high-income status. The demand for animal protein continues to grow. And so there's probably lots of solutions, but one of the big grand challenge solution is to try to enhance the efficiency by which livestock produce products for human consumption. And so what I mean by livestock production efficiency is how well animals can take the inputs, such as water, both the amount and the quality of water, disease prevention, various strategies, including use of antibiotics, feedstuffs, both the quality and the quantity of feed that is put into the animal, and how well they can convert these inputs into outputs for human consumption, like meat, milk, and fiber, defines their production efficiency. And the one intrinsic element within the animal that can influence how well they can convert inputs to outputs is their genome. And so by selective breeding, we can have impacts on shaping the genome, and this can have positive impact on genetic gain, the improvement in traits or performance from generation to generation through selective use of genetics. But there's also limitations and challenges to selective breeding. It's based primarily on an observable phenotype, and that makes it somewhat messy. It can take 5+ generations and 10+ years to make incremental changes in traits, and genetic drift and inadvertent selection of negative phenotypes can happen. And there's plenty of examples in the livestock world of negative phenotypes arising from recessive mutations that were inadvertently selected for. And so really the promise of gene editing is that we can combine molecular tools with the gobs of genomic DNA information that have been produced for animals for the past 20 to 30 years, and combine those two things to achieve efficient and precise genetic engineering, and do this in 1 to 2 generations, and clean up some of the messiness that comes with selective breeding, and also speed up the process by which changes can be made in trait development. And so CRISPR has really arisen as the gene editing tool in the toolbox for creating genetically enhanced food animals to address the future of food security. And I define the leading edge of gene editing in livestock for the future of food security in 4 major bins. The first being welfare, improving the health and well-being of the animals. Resiliency, in terms of being able to be resistant to diseases in a variety of settings. Efficiency and growth, how well animals can add muscle mass and palatable muscle mass to the skeleton. And then reproductive efficiency, devising of applications and tools by which we can get dissemination of trait-driving genetics through reproduction. And so I'm going to spend the rest of my minutes talking about reproductive technologies because they can increase access to trait-driving genetics based on the simple principle that all generations are linked through their germ cells. The germ cells are the conduit by which genetic information flows across generations. And so to improve traits within a population, it must come through the germline. And so technologies that can achieve widespread dissemination of gametes, either in the form of sperm, eggs, or the embryos that they can make, if those are made widely available, they can have dramatic impacts on genetic improvement within populations. One example of a reproductive technology that has had dramatic impact on livestock populations is artificial insemination. And that is because we can take a bull that exists somewhere, say in North America, that is deemed to be of elite or high value genetics, collect the sperm from that male, put it in a cryopreserved state, and ship it around the world for artificial deposit in the reproductive tract of females that are in the right stage of their estrus cycle. This is something that has commercial origins back in the 1940s and 1950s. And one of the industries that we can see dramatic impact of artificial insemination is dairy cattle production. And so this is the pounds of milk produced per cow per day for Holstein breed cattle, in which roughly 80% of all animals are bred by artificial insemination. And it's been more than a quadrupling since the incorporation of artificial insemination as a breeding tool. And so it's this increased access to genetics that has really driven a lot of this increase in production efficiency. And so although artificial insemination and the ability to cryopreserve sperm provides a means to disseminate elite genetics on a widespread scale, there are problems. The first is that in beef cattle, it's a logistical challenge to effectively apply artificial insemination. Producers need to be able to track the reproductive cyclicity of females and need to have a skill in depositing the sperm in the reproductive tract at the right time in the right place. The majority of beef cattle production around the world happens in range or pasture-based conditions where producers only see their animals once or twice a year. And so the logistics of applying artificial insemination don't work. In swine, cryopreservation of sperm is inefficient. And so although there's a lot of artificial insemination used in swine operations, it's with animals, males that are on site or in a regional context. And so the global reach, the global access to genetics is somewhat limited. In small ruminants like sheep and goats, it's a technical challenge. This is largely artificial insemination is largely done through a surgical procedure, and most producers do not have technical capacity or the access to equipment to be able to use this. And so if we look at the percent of animals that are bred by artificial insemination in the dairy cattle, beef cattle, sheep, and goat populations around the world, roughly 80% of all dairy cattle are bred by artificial insemination. Only about 7% of beef cattle globally are bred by artificial insemination, and less than 1% of sheep and goats are bred. So there's been lost opportunity to access elite driving, trade driving genetics in beef cattle and small ruminant production. So we think one of the solutions is to try to engineer surrogate males that are going to produce genetically desirable sperm, and they're going to be able to disseminate that by natural mating. All the benefits of all the genetic benefits of artificial insemination, but not having to use the artificial part. And so our approach has been to develop a technology we refer to as surrogate sires. We believe that this can be a next generation livestock breeding tool that has application across the spectrum of livestock species. I'm going to use cattle to display the concept of what a surrogate sires breeding program can be, but you can plug in almost any livestock species in any scenario into this. And so the idea here is that in beef cattle production, breeds like Angus that reside in North America and other places in high income countries have been developed for their carcass characteristics. So the palatability, the tenderness of the meat product. Yet the majority of our beef cattle production globally happens in South America, and the breed of choice in South America is Nelore cattle. Nelore cattle have been selected and bred for resistance and resiliency to the South American climates, but the carcass characteristics don't meet those of Angus cattle. And so if we want to bring Angus genetics into Nelore populations to increase the carcass quality and growth efficiency, one idea would be to take this bull and place him in South America with a population of females and let him breed by natural mating. This has been attempted. These bulls typically last a few months. They're not adapted for those environments. The second is perhaps you could take sperm from these animals, from the Angus donor, take it to South America and use artificial insemination. Again, the majority of beef cattle production, including in South America, is done in a range-based or pasture-based condition. And so there's been some improvement, and I heard a couple of months ago that it's moved from about 6% to 10% use in places like Brazil, but it's still the minority of cattle are bred by artificial insemination in South America. And so we think one way to achieve this is we can collect stem cells, sperm producing stem cells from this Angus donor, amplify them in a culture dish, and transfer them into Nelore recipient bulls that can breed by natural mating and pass on genetics. And so these recipients really need to be germline ablated and not be able to produce their own sperm. And so we've been working on this question for many years of how do you generate male livestock that don't make their own sperm? And it wasn't until CRISPRs came on the scene and were available from mammalian cells that we could get after this. And that was to design a strategy to knock out key genes that are needed for germ cell formation, but would leave the testes intact. And in that way, we could create sterile surrogates with functional testes for stem cell transplantation. And so we went after this gene nanos2. We did this because we knew from model organisms that the nanos genes are required for germ cell formation. In mammals, there's 3 nanos genes, 1, 2, and 3. Knockout of nanos1 does not produce a phenotype. Nanos2, knockout of nanos3 in a rodent model makes male and female sterile, but knockout of nanos2 produces a male sterile only phenotype. So we adapted a CRISPR-Cas9 gene editing system and applied it across cattle, pigs, small ruminants, and repeated studies in mice to show that CRISPR-Cas9 editing of the nanos2 gene to inactivate it could make a male specific sterility phenotype. And so we've been able to show now that we can transfer sperm producing stem cells in the testes of nanos2 knockout males in pigs, cattle, and goats and achieve donor derived sperm production. And so we do think that we're at a point where we're refining the procedure and working with partners across the globe to bring this technology to bear on livestock production in multiple types of animals and in multiple regions of the world to achieve widespread dissemination of elite genetics in a natural breeding scheme. So considering that I'm running out of time, I won't spend time talking about my thoughts on welfare, safety, and how surrogate sires can be regulated, but I'm happy to address any of those questions in the Q&A session. And I'll just leave it by saying, giving my perspective on what a GMO trigger should be for food animals. And I think that should be that there is a distinction between animals that have incorporation of foreign DNA into the genome that could not occur without human intervention. And that would be defined as a GMO. But animals with genome edits that are changes in DNA that could arise in nature, such as indel mutations or polymorphisms brought about by CRISPR-Cas technology, should not be considered or regulated as a GMO. So with that, I'll say thanks again for the attention, the opportunity to provide my perspectives in some of our science. And I'll answer questions, or I'll hand the Zoom podium over to Professor McGrew.
Thank you very much, Sean, for this nice presentation. And let's see if we have any questions for you, but while we wait for them, I was going to ask about the regulation of these animals. I can see you didn't have time to talk about that, but I'm going to ask you about that and how easy it will be to have these animals on the floor soon, or how do you envisage the regulation in your country and in our countries, in South America and the rest of the world?
Perfect. Well, maybe that'll allow me to go back to my slide.
Yes, please.
So I'll get my slide through your question and answer. So I think that the big things, and Mike McGrew and I were at a workshop in September put on by the USDA Foreign Ag Service in Brazil about the regulation of biotechnologies in food animals. And so we heard a lot about some countries where they're already making decisions. And largely in the countries that are approving gene-edited food animals right now, it is that distinction of whether there's foreign DNA in the genome or not. And so gene editing technologies that bring about just insertion, deletion mutations, or even small polymorphisms that can arise in nature are being regulated, or are not being regulated as a GMO. And I hope that's where the US ends up, but I can't wave my magic wand with that. But there's two big things. One is the welfare concern and then the safety concern. The welfare is that the gene editing that's being done doesn't decrease the welfare of the animal. And I think when it comes to food animals and developing a gene editing application that is going to go into production settings for making food animals, that you can have this unanticipated negative phenotypes could arise in an R&D phase. Yeah. But through careful design and beta testing in model organisms, those things limit themselves out. And they do that because when you move into a commercial side and in a societal public impact side, I don't see any scenario where a producer is going to take forward a gene editing application that reduces the health and efficiency of the animal. And in that way, it's self-regulated. These welfare concerns, I think, can be self-regulated, and they can be regulated through management. The safety side of things, again, I think there needs to be a distinction of edits and DNA changes that are brought about by gene editing in terms of indels and polymorphisms, because these can arise during just natural mutagenesis that happens through cell division, double-strand break repair, meiotic recombination. These are all things that could arise in nature. And so the safety thing, I think, needs to be reconsidered on what the difference is between a CRISPR-modified, a CRISPR-derived mutation and just normal mutagenesis that happens in the mammalian genome. And then the off-target edits situation probably needs to be looked at some level, but random mutagenesis during cell divisions also creates just random off-target, changes. And how can we really distinguish these when we don't have inbred lines of cattle to compare to? And is it necessary to really address these things? And so I think that where the world is going, where I hope the world ends up, is that gene edits that could arise in nature, and that would include insertion, deletion mutations, and polymorphisms that are already found in animal genomes, are not going to be regulated as a GMO. But anything that involves the insertion of foreign DNA into the genome is going to be regulated at some level.
As OGM. Yes, at least here in South America, the discussion is going that way also. So thank you very much, Sean, for your considerations. And I don't see any more questions in the panel, so we can move to the next speaker, who is Mike McGrew. We are going to change species here. And Mike is, let me introduce him. He's personal chair in avian reproductive technologies at the Roslin Institute in UK. He joined the institute in 2001 to help develop transgenesis in chickens for use in developmental biology and biofarming. His laboratory works on primordial germ cells in birds to generate gene-edited chickens and to create biobanks using germ cells. And he will talk today about how genome editing can be used to improve resilience in poultry and preserve genetic diversity. So thank you, Mike, and we are happy to listen to you.
Thank you, Martina. Let me put my slides up. There we go. Okay, sorry, I can't see you in the video. Can you see my slides and hear me okay?
They are okay, yes.
Yeah, okay.
You can go ahead.
Yeah, thank you. Thank you for the invitation to speak to you today about my research. And it's nice to go after John Oakley to present, because he gave such a nice introduction to where we are in genome editing and livestock. So what I'm going to talk to you about today is how we can use genome editing to improve both resilience and conserve biodiversity in poultry species. And as John told you, the potential of genome editing in animals is enormous. And this is because we have the ability to improve any trait that's genetically based. And this isn't limited to existing genetic variation. We can introduce the genetic variation that we want into a particular species. Now, this has been done for many livestock species now. One of the most recent cases, here, I want to turn, just 1 second, I'll turn on my pointer. One of the recent cases is introducing the myostatin mutation into sea bream in Japan. Here at Roslin, we were part of producing the PRRS virus-resistant pigs. It was a single edit that made these pigs completely resistant to avian influenza. The hornless cattle, the polled allele, has been introduced into cattle, which leads to increased welfare for both the cow and for the farmer, because there's less injury from the horns during standard husbandry. And I should say the first disease-resistant chickens were produced by my colleagues in the Czech Republic and in Germany. And these were edited to have disease resistance to avian leukosis virus. So that's a virus in poultry that we don't have a vaccine for, and these chickens were edited to have complete resistance. Unfortunately, a picture of the chicken isn't online, so I had to show you a cartoon caricature here. So what I want to convince you of today is chicken is probably the most abundant and probably the most important livestock species. So this is the data for 2020. This is, of course, the year of COVID. And if we look at the number of animals slaughtered, this is globally, you see there's a huge number of animals slaughtered. But if we look at this, the vast majority of animals that were slaughtered for food in the world is chickens. And just in 2020, over 70 billion chickens were slaughtered. This was both for beef production, for meat production and for egg production. So this is why chicken is the most, I believe, the most important livestock species on the planet. So chicken, of course, is not a mammal. And as such, both genome editing and cryopreservation has been extremely challenging in avian species. And this is showing the oviduct of the animal. And this is showing the oocyte here, the female germ cell. And as you probably know, it is extremely large and it has a huge amount of yolk present. And also when the egg is laid, it is for chicken is 24 hours old and has developed to 60,000 cells. So this makes the manipulation of the oocyte very difficult. And also it makes cryopreservation of both the oocyte and the egg impossible because we simply cannot freeze this down and thaw it out. The yolk makes this impossible. And also cryopreservation of semen is very difficult in avian species. It would work for one breed of chicken, yet in the next breed of chicken, it's very difficult. So one advantage we have in poultry is that you don't have to sacrifice the mother to get the egg. So the chicken will lay, the female will lay an egg every day. The egg is 24 hours and starting to develop when it is first laid. And if you want to access the embryo for genome editing or for other manipulations, you can simply cut a hole in the shell here. And this is showing you a chicken embryo at 3 days of development. So at this stage, the nervous system hasn't formed. So you can simply go through, open the shell, do your manipulation. You can simply tape the shell shut and culture this chicken to hatching, which will occur at 21 days. And we've been interested, or lots of labs are interested in studying how the embryo forms using the chicken embryo. On the right side, we show you a embryo or a chicken that was genetically engineered to carry the green fluorescent protein. And we did this so we could actually observe the embryo developing during the early stages of development just by shining the fluorescent light on it. And here you can see the chicken embryo developing. Its picture is taken every 5 minutes over the first 6 days. We're able to just image this embryo in vitro in the egg and study its development. So genome editing is a bit different in avian species. And what we tend to use, John showed you that, and I think Angelica showed you injecting of these CRISPR tools in the early embryo. Well, again, that's very difficult to do in chicken because the laid chicken egg is highly developed. So what we simply do is we edit the early germ cells, the primordial germ cells for chicken. So what we can do is these actually migrate through the blood system of the early embryo. So we can simply go in, sample the blood from this embryo and put these cells in culture. And we actually came up with a very robust, defined culture medium to grow these cells. And I should say, this is one of the rare animal species for which we can grow or culture primordial germ cells. So these will proliferate in culture. We can then cryopreserve them and put them in the freezer to use at a different stage. And this is showing you, the cells in culture. And this is showing the manipulation of how we isolate these cells. So this is a day three chicken embryo. We simply go in with a glass pipette here. And again, the embryo hasn't formed a nervous system, so it is welfare friendly, and we simply take a microliter of blood. And there we are, and that is the material we put into culture. So at this case, we can take our CRISPR, Cas9, or TALEN tools and transfect them into the germ cells, and then we can select for the genetic change that we're interested in. And then to make these develop, we have to inject them into what we call our surrogate hosts. This is similar to what John said. So this will carry the exogenous germ cells, which will contribute to its own germ lineage. And again, we tape this egg shut, we raise it to hatching, and we breed from it. In this case, we put a GFP gene into the primordial germ cell, and some of the offspring came from this germ cell, as you can see from the fluorescence. So this whole process, I should say, is very inefficient. The germ cells that are introduced have to compete with endogenous germ cells. So like John, we thought of making a sterile surrogate host using genome editing to actually increase the germline transmission. So we did this in two ways. We first used genome editing to delete a gene that was important for germ cells to develop. This is called the DDX4 gene, and we published this several years ago. So the females in this case have no germ cells. So if we inject germ cells into a surrogate host female, we get 100% germline transmission. We also came up with a transgenic surrogate host. So this actually contains a transgene. In this case, we put a transgene that introduces cell death, and we genome edited or engineered the genome so that we express this protein only in the germ cells of the animal. Then when we gave this is called the ICAS-base9 gene, and then when we simply give it a neutral compound, it will activate the gene and ablate the endogenous germ cells. So this is our surrogate system to create genome edited animals, and we were the first to use it using sterile hosts, but it's being taken up by other laboratories around the world. So simply what we do is we isolate germ cells or the primitive germ cells from any breed of chicken. We can put these into culture, and then we can genome edit them and then select for our change that we're interested in. We then will inject these back into a surrogate host along with this BB compound. We can then raise these animals to sexual maturity and breed them to produce our genome edited offspring. And the important thing is we can do this for any breed of chickens. And this whole process takes 6 months from injecting of the germ cells to producing offspring. And this is just showing you how we inject these germ cells and this BB compound back into the vascular system. So it's at the same stage that we actually sampled the blood, and we add a green dye so we can see our injection. And you can see it's harmless to the early embryo. We simply inject a few thousand of these germ cells. We then seal it up with a piece of tape, and then we hatch this surrogate host. We will get 100% transmission of this offspring. So one proof of principle, we targeted a gene that gave white feathers to a chicken. It's a dominant natural mutation. And we simply repaired the mutation in the genome of chickens having white feathers. And this is showing the result here. And the chicken on the right has this mutation that gives it white feathers, and the chicken on the left has been edited and is homozygous for the repairing this natural mutation. So you can see the technology is very robust, and it's high welfare friendly. So in my remaining 5 minutes, I wanted to talk about 2 different applications that I would say would be useful for using genome editing in poultry. I showed you one that was done by my colleagues. Also, this was to edit chicken to make them disease resistant. This was for avian leukosis virus. I think a really important target would be to edit a chicken for disease resistance to bird flu. As you know, in Europe and all over the world, we're having immense problems with H5N1 being transmitted in wild type birds to domesticated chicken, and is leading to their huge amount of disease and of culling in this chicken. And the big fear we have is somehow the disease that once it moves to chicken, it will be able to mutate and then move back to humans and start a global pandemic. So this is a very important problem, and probably one of the best ones we could solve, hopefully using genome editing technology. So as I said, genome editing through mutation of a viral receptor has already been shown, and this has been done in pigs, and this has been done in chickens, and this has also been done for the PERS receptor, CD163, by my colleagues at the Roslin Institute, and also, to engineer disease resistance into swine. So for avian influenza, it's a bit more difficult. There is not, the receptor for avian influenza is expressed on many cells, and it's also needed, it's actually needed that we wouldn't be able to knock it out. Transgenic chickens that suppress the transmission of avian influenza were generated by my colleagues here at Roslin and also colleagues down at London. And we and others have come up with this strategy to engineer avian influenza, and this is to identify host factors present in chicken that are needed for IAV replication, and we would gene edit these host factors so they would no longer be able to support avian influenza replication. We would produce these chickens, and then we would be able to challenge them and hopefully propagate this allele into other populations of chicken. Now, I can't show you the results from these experiments because they're still in progress, but our preliminary findings are that we will need to edit many genetic targets to be able to have complete resistance to avian influenza. So this would mean multiple genes would be needed to be targeted in a poultry to be able to have resistance. And I want to show you this online public dialogue that was carried out by the Nuffield Council on the use of genome editing in farmed animals. And one of their conclusions was that a single use of genome editing one allele, one change, would be acceptable, or might be more acceptable for human consumptions rather than engineering multiple edits into a chicken. So this is going to be a problem if we need to edit multiple changes in the genome. It'll be less likely to be accepted by the public. The second example I want to show you in my last 2 minutes, I hope, is about conserving avian biodiversity using gene editing. As you might not know, there are actually thousands or hundreds of chicken breeds that exist across the planet, over 1,400, and in many different countries, in every country around the world. Most of these are used, these are indigenous breeds that are used for local production. We also have very many selected breeds for commercial production, which are very highly productive and emit lower greenhouse gases. The idea is that we wanted to develop a technology to be able to biobank these, because as I said, it's very difficult to freeze these chickens down and bring them back. So this is work done by June Hugh in my laboratory. And simply what we do is we take the reproductive material from these early chicken embryos and we put them straight into the freezer. We don't even bother culturing the germ cells. And then at a later stage, we can take this material out, we inject it into our sterile surrogate host, we raise them to hatching, and we breed from them. So simply in one step, we go from frozen germ cells to reviving a chicken breed. And this here shows the power of this technology. In the back, we have our iCas-Base9 sterile surrogate hosts. These are genetically modified. And in the front here, we have the offspring from a rare breed of chickens, or a rare commercial breed of chickens here. They have the black skin, and genetically, they're not related at all to the surrogate host. So this is, if I can go on, this is mostly to address the benefits and harms of genome editing. This can be part of the discussion session. But what I want to point out is the benefits and harm of genome editing really depend on your position of raising livestock for meat and food products. And we can't begin to have the argument if genome editing would be beneficial or harmful with people who think that livestock should not be used for meat or food products, because it will negate our arguments on both sides. The first thing is the potential benefits I see is that we can increase increase food security. We can produce more robust animals, and these can also be disease resistant. In terms of animal welfare, we can reduce the number of animals dying from disease, and we can also replace harmful practices. This can be dehorning of cattle. We can also improve the environment. The chicken will need less feed. They will produce less greenhouse gas and waste products. And also a further thing to consider is they can improve human health. We could need to use less antibiotics to treat animals. On the other side of this is if you're against farming of livestock animals, all these benefits lead to increased harms. So in terms of food security, we're increasing the use of specific breeds of chicken that were edited. We're losing biodiversity. In terms of animal welfare, we're not completely eliminating animals using rearing. We're still going to raise animals for food production and for meat production, but these animals will be healthy, but they still will be raised. And also in terms of the environment, we're not eliminating the impact of animals on the environment because we will still be farming animals. And the final point is that in terms of human health, we will be promoting the use of meat production or meat in diets. So I'll stop there.
We will have to finish, yes.
There, I will thank my funders and the people who did this work. And thank you for your attention.
Thank you, Mike. I think we will have more time to discuss all the things later in the roundtable. We have time for one quick question that we have in the panel that I'm going to read it. And it says from, Anonymous. You mentioned multiple gene edits would be needed to get a completely influence-resistant chicken.
Yeah.
Roughly, how many will this be? It is viable?
Yeah, so sorry, that was my hypothesis that we will need multiple edits. And also we will need multiple edits that don't, if we do this, that don't impinge on the health of the animal. Yes.
Okay. Thank you. So I think all these examples are great science, and I'm worried also about the regulation of all this and when are we going to see all this application on the field? And I think we can again discuss this in the next roundtable. So thank you both, and we move to the next one. Keep in touch.
Good afternoon, everybody, and thank you very much, Martina, for providing us with a very interesting two papers. It's interesting, if I may, before I start this, to say I was very involved in both the setting up of the Cartagena Protocol on Biosafety and of working with developing countries in setting up their law in relation to that. And one of the things which we meant when we wrote that protocol was that if the modification could be achieved naturally, it was not a GMO. It's already in the international agreement. And I thought, because of what was said earlier, I felt that was necessary to say. That's what we thought we were writing. If people haven't read it that way, I'm sorry. We're going to have a roundtable now on the social and ethical implications of genome editing in animals in particular. Again, may I remind you that the European Group on Ethics in Science and New Technologies produced a report, an opinion to the Commission and to the Council of the European Union exactly a year ago on genome editing and its ethical implications, and that has a very significant section on animals, and I can refer you to that. Moving on, is Andy Greenfield with us? Because I haven't seen him on the list. And he's supposed to be talking at this moment from 3:30 to 3:50 p.m. Andy, are you here? I suspected not, because I can't see him on the list. Can I move to Christine Tate-Burkhardt, again from the Roslin Institute, to address us on this topic for about 20 minutes?
Yes, let me just put on my camera and get me off mute. There we go. And I cannot even see myself. Here we go. All right. So let me share my screen. Here we go. So I presume it's coming. There we go. Now you can see this. Brilliant. And you don't need to see yourselves. Here we go. Sorry about that. So we've heard an awful lot about genome-edited animals, and because I come from the genome editing side as well, I shall talk again about somewhat genome editing in animals. But as we heard, or as we already read in the outline for today's roundtable, when we talk about animals, we can talk about many different things. That includes managed animals, such as bees, who are now subject to genome editing. But we also have heard lots of discussion about editing in companion animals, such as cats, where there's discussion about editing the FeLD1 protein, which is responsible for allergies. So I have specifically excluded them from this presentation, but of course there's many social and ethical implications that arise from that. But today, let's go back to what we define as genome engineering, what we define as genome editing or genetic modification. In principle, genome engineering and genetic modification are almost replaceable, but it's basically the direct manipulation of an organism's genome using biotechnology. And those can both be old technologies, such as transposons, but also new genome editors. And what they can achieve in the end, sorry, I'm going the wrong way here because I'm on my home computer. They can insert new DNA into a host genome. That can be transgenesis or cisgenesis. We can remove genes or knock them out, and we can do subtle modifications and introduce minor changes, which may be new genetic codes, or again, they can basically mimic cis or transgenic elements or be completely novel. Now, surprisingly, I'm going to start with transgenesis, because we had decided already years ago that transgenesis, moving human genes into primarily goats and sheep for production of human medicines, was acceptable to us ethically. From maybe a more utilitarian or a contractarian view, we decided that making animals transgenic for the production of human medicines was acceptable and approved by the EMA and later also by the FDA. And this is probably because we have a history of using animals for the production of human medicines, whether that is insulin from horses or drugs, antibodies for diphtheria. We grew up with that, and we accept that medicines can both be transgenic, genome edited, and they go into the human body similar to how food stuff do, but we have different perceptions of risk when we look at those different uses. Again, whether that is a risk-benefit analysis, and we are more happy to risk something when it's for our own health, because the alternative is to be sick versus eating something, whereas the alternative is to eat something else, is maybe a different risk perception. But now let's go into some examples that we actually have with genome editing. We can do disruptions, deletions, modifications. Why is that important? What do we use genome editing for? Well, if we just look at a livestock breeding side of things, we can, for instance, have animals or traits, whether that is disease resistance, that's where the slide is coming from, that are not present at very large amounts in our breeding stock. And if we would breed these animals for selection because we want them to have resilience to specific disease or as John said, the slightly bent back here or some other traits, we risk inbreeding, we can risk productivity loss, or we can risk losing other traits. But that is probably a minor application strategy. It's more when we're starting to look at traits that are either present in indigenous breeds with completely different traits, different fur color, different sizes of litter, etc. So we're facing strong productivity loss and the loss of other traits. And then when we're looking at cisgenesis or complete integration of new traits or deletions that come from lab research, this is, of course, not possible through natural breeding necessarily, because inbreeding between or breeding between natural species may lead to either a sterile animal or it is simply biologically not possible. That's the biology behind it. But what is the application behind why we want to do genome editing? Well, we are all facing big challenges. We have massive population growth and we need to feed the world. We see the developing world increasing in demand for animal protein, which is obviously contrast to trying to do sustainable feeding of the world. We have climate change at the same time, but also the environmental impact of growing animal protein in itself. And the cattle are obviously affected by heat stress behind here, but they're also the ones that produce, for instance, a lot of methane. We're trying to reduce antimicrobial use and improve animal welfare, which is again in contrast to having to feed the world and increase agricultural production. But we also have food waste in the production chain, whether that is through disease, overproduction in the wrong places, etc. So, some examples of what has been done. First example is our own pigs, which have been mentioned before today. It is resistance to porcine reproductive and respiratory syndrome virus, a viral infection that affects primarily suckling piglets, where it can lead to up to 100% lethality due to diarrhea and systemic disease. But also it affects severely pregnant sows, where it can lead to complete abortion and death of fetuses in utero. It affects all pigs through reduction of growth and systemic disease. And more importantly, it also affects them systemically to make them more susceptible to other disease. So we have animal suffering, we have the loss of animals and growth and food waste in the production chain, and we have a viral infection that incapacitates the immune system. This leaves the door open to secondary bacterial infections or other pathogens, which increases the use of antibiotics. It's a global disease problem, and consequently, this is also obviously a financial problem. What we have done is looking at the biology of how this virus enters the cell, and it uses a specific key lock interaction to actually gain access into the cell rather than getting destroyed by our cell's partial immune system. And the way that the virus interacts is through interaction with this protein, CD163. It's like 9 pearls on a string. And the virus interacts solely with bead number 5 that doesn't have biological functions. So consequently, we can remove bead number 5, make these animals through transgenesis, test them in vitro, of course, to a large extent before subjecting some animals in a controlled fashion to disease. And these animals were found resistant to PRRS infection. Now we have animals with improved animal welfare because they're not getting sick, they're not getting a systemic illness, they're not susceptible to secondary bacterial infections because they don't get the primary infection. We're using less antibiotics. We have pigs that no longer shed and pigs that are not transgenic. So we have a happy animal. We can shield other non-resistant farms. So in principle, this is a win-win-win. We also see economic progress of these animals. They're going through FDA approval, and this is expected to be complete sometime in 2023 and 2024. But from a why is this useful perspective, these animals can reduce food waste, they can reduce antimicrobial use, they can help feed the planet and development, and they improve animal welfare. So there's a lot of benefits to these animals. What are the risks of these animals? Is it off-target effect? Are we worried about off-target effects? How do we regulate intense farming? Lots of questions that are linked to this. Another example, we have heat, we have an increased heat of the planet, climate change, and that can impact animals of all types, but I'm talking about cattle today because that's the example that is the furthest advanced. In Scotland, it's not really a problem. We have 2 seasons, it's June and winter, but for the rest of the globe, it might actually be a bit more problematic. And actually, to be honest, it's also becoming exactly the same problem in Scotland, that you have increased heat. This affects cattle's metabolism, there's reproductive failure, increased occurrence of mastitis, and reduced food intake. So consequently, we have less production and we have natural solutions. And here, this is a slick phenotype Xenopol cattle. You see it has a shorter hair coat compared to this non-slick phenotype cow. And they are able to maintain a much lower core body temperature and less affected by heat stress. This can also be, and this is the basis of this is a change or a truncation in the prolactin receptor that doesn't only affect the fur coat, but it also affects signaling of stress signals in the cattle. So it has a systemic impact. We can make these cattle with genome editing on different genetic backgrounds, such as the Angus cattle seen here, but they are also naturally occurring primarily around the Caribbean Sea, Central and South America. Xenopol cattle, Carora cattle, and Criollo Limonero cattle all have this slick genotype. So consequently, through to their natural occurrence, FDA has already deemed them as low-risk genome-edited animals, and in Brazil, they're approved as conventional cattle because they could be occurring through natural breeding. But once again, we have a lot of advantages here, as again, we are targeting big questions of population growth, development, climate change, and animal welfare. So again, do we have ethical questions here to rather, do we impact the welfare of animals by not introducing these changes through genome editing? Is it our responsibility to do this? What is the risk of introducing this? If we look at animal welfare, and I'm sorry for the pictures that are to come, but sadly they have to be shown. In Europe, we do dehorning often at an early stage and in an early age following sedation of some sort or description. But in a lot of countries around the globe, dehorning has to be carried out at a later stage of development, which means a very painful and bloody procedure. So here again, we know that some cattle breeds are naturally hornless and they have a small duplication in the POLT gene, and that leads to animals being naturally hornless. We can breed these into cattle and improve animal welfare immediately. Again, should we do this? Is it responsible to do this? So let's move on to production trait, and probably one of the most contested questions around genome editing livestock, because we look at this when we look at European breed. Bos Taurus, we have 2 natural double muscle phenotypes, so myostatin changes to Piemontese and the Belgian blues. We have other ones. We have a microRNA in Texel sheep that also changes the expression of myostatin, and they're all double muscled. We also know that they have welfare issues because, for instance, breeding with these animals becomes much more difficult and they're much more likely to have cesarean sections. However, that is on the basis of highly bred European breeds. If we look at Bos indicus, for instance, it actually brings the animals on a similar productive level as normal Bos taurus. So question is, are we impeding development by saying this is per se negative, or should we look at developing countries where different animal breeds are more prevalent? And then, of course, we have animals where the double muscle phenotype doesn't impact a birth canal, namely fish. And of course, that is where this double muscle phenotype, which is a small deletion, has been implemented and has been often deemed as conventional. We have the double muscle tilapia in Argentina. We have the red sea bream and the tiger pufferfish, all double muscle genotypes and all approved in different countries around the world without any specific labeling. Now, here we stand at the crossroads between developing the world, feeding the world, but also negating that with, or leveling that with animal welfare. And that is where we talk about genome editing, but we shouldn't forget that transgenesis has a lot of advantages too that we could benefit from. We all know the AquAdvantage salmon, which is now approved in the US and in Canada. It is a transgenic animal with a promoter from the ocean pout, a cisgene integration from Chinook salmon. But basically what it does to these salmon is that rather than just growing in spring and summer, these salmon now grow all year round, because in captivity, there is food available all year round. So suddenly we get a much earlier target weight just by adapting to a growth system that is in our normal intense farming environment. But there's, of course, large questions around aquaculture here, because especially salmon get reared in environments where escape of these fish, if they're fully fertile to nature, could impact naturally occurring populations. So again, the questions, how do we regulate this? Because it's much easier to contain a pig, a chicken, or a cow, and there's also less risk of them inbreeding with wild populations, but how do we deal with fish? Well, in the case of AquaBounty, they're landlocked, highly controlled facilities, but we have fantastic research from Norway showing sterile salmon as a true opportunity to actually mitigate these risks. And last but not least, an older example of transgenesis, where we have the EnviroPig, which allows us to reduce the phosphate excreted by pigs. As we know, phosphate gets taken up from feed, and it cannot get processed by pigs. One of the opportunities we see is the reduction of phosphate-rich food in pork production. But there's an interesting example in Europe at the moment where some countries like Switzerland have introduced lower levels of phosphate permitted in pork feed. But what is happening is that this is fine for commercial production, for traditional commercial production, but the low phosphate feed reduces productivity in organic farming. Consequently, what has happened is that the phosphate levels for conventional farming have been lowered, yet it is permitted to use higher levels of phosphate in organic farming. So that kind of defeats part of the purpose of organic farming. And we have to keep these discrepancies in mind. Here we have a transgenic solution to this, where we have a transgenic animal that contains an E. coli, so an E. coli protein enzyme that can break down phosphate and eventually leads to the lower production of phosphate in pig excrements. So we have less pollution of rivers and lakes and eventually the sea. So lots of examples, and I'm sad to say more questions than answers. And I just leave you with a few thoughts. We have one side of the medal, which is regulation. How do we regulate? We need to regulate because no regulation is worse than a tough regulation. How should we regulate traits rather than technology? Probably many of us are on that side. How are edited animals recognized and checked? And actually the European Union is launching a lot of funding or a part funding this year to actually do exactly this. How do we recognize genome-edited plants and animals? And in some cases, we actually have to say it's technically not possible. It's an association by proxy. It is more likely that an animal has been generated by genome editing rather than natural breeding, but it is not impossible to generate that animal through natural breeding. How would that hold up in court? And how does it impact trade? On the other hand, we have animal welfare and intense farming. How do we regulate that? Does genome editing actually play into this? Or should animal welfare and intense farming be regulated elsewhere? We have a changing world. Climate change as well as population development is a problem. That means we have closer contact to livestock. Zoonotic disease is one consequence for us humans. But we also have a developing world where demand for animal protein is high, population density is often high, and the risk of zoonotic disease is high. Apologies again. Do we have a social responsibility to roll out animals in these countries? How do we do that? How is that regulated from a prospect of patenting? How do we make money? Because it's currently not cheap to make transgenic animals. And how do you eventually permit sustainable production for all? Lots of questions, and I'm afraid I don't have all the answers, but exactly that's why we have the round table. So I'm just going to thank all the people whose work I've presented here. I've not named everyone with names, otherwise you couldn't read this anymore. And I also acknowledge my funding.
Thank you very much, Christine. It was fascinating when the European Group on Ethics had to look at these issues, because the Commission specifically asked us to look at non-human primates, which we haven't talked about, which is primarily humanization, I guess, they thought of. We've commented that the 3 R's aren't sufficient to address many of the issues that you've raised in the last slide particularly. But we did feel very strongly that the technology should be used.
Yeah, I specifically didn't touch on the human transgenesis because I thought I would touch on that. It's also not my specific area, but I'm sure Angelica can weigh in on this as well.
Absolutely. At the moment, I have one question from Jane Lampram, which is, can cattle be gene edited to decrease their methane production? I'm not sure.
I know it has been I'm quite certain it has been done in pigs again as well. It's again a transgenic solution. So the transgenic solutions, of course, exist because we have lots of bacteria who can break down methane. So there is usually a relatively straightforward approach of using transgenesis. The question is, are we happy to use it? how do we use it in organic production? Because I think that is exactly the problems that we have. We think about natural, despite basically having not a single natural animal in livestock production, whether that's organic or conventional. But at the same time, of course, transgenesis is another question. But then we look at the US and we have transgenic crops for decades and no negative effects to human health. Of course, questions with monoculture, pesticide use, etc. But for an animal where this protein would just be used to break down methane, it's a different question, in my opinion.
Thanks for that. Hopefully, Jane, we've answered your question. It can be done. We have to think about whether it should be and what the implications are. I have no further questions, but I can comment about a number of things if I might. Jane has said thank you. When we were talking about this in the European Group on Ethics, we were being asked, and you've mentioned this in your talk, about can we identify those animals which have been gene edited and separate them logically from those that have not? Well, there were two things we addressed there. One was that even if we regulated this in the European Union, we couldn't control what happened elsewhere. And unless markers were required by everybody, some kind of marker when you do make a change, then it would be extremely difficult to identify animals which had been changed without any form of marker. So regulation, if it comes in at all, would have to be an international agreement. It couldn't be done just by saying the opinion, we won't have gene-edited animals, because that's not going to work if we import meat or eggs or fish or whatever. So we were aware of this and did attempt to suggest to the European Union that there was a real problem of regulation if we wanted to do this. Further question that arose was multiple traits being inserted, which changed the animal very significantly. One of the projects which had come to our attention was somebody who was trying, this was in plants, but somebody who was trying to change the response of plants to light by changing the range of electromagnetic radiation to which they responded. Okay, that's a huge change. It requires an enormous amount of genes being inserted into the plant. Now, I'm not sure we're going to insert photosynthetic genes into animals, but this is the sort of thing which we try to think about, and the implications for animal health, animal welfare, human welfare, and environmental impact, all of which we need to think about. I know I'm throwing it to you, Christine, but it's open to anybody else in the room, which is about 80 people, to comment and think about.
Yeah, I've had exactly that discussion. I've been talking to the European Union GMO reference labs recently, and that is exactly the question. So on one hand, we want natural animals, but on the other hand, to have a natural animal that we can, or a natural-like animal that has been generated through genome editing, in order to trace it, we have to put in a marker. So it becomes less natural again. And this is kind of the juxtaposition where we're at. And the reality is, if we look at the approval of slick cattle in Brazil, they have been approved as conventional. They will not be labeled as genome edited. We know how much beef we actually import in Europe from Brazil. That means genome edited animals will be on our tables. And the reality is, because this is a natural like mutation, we cannot trace this. Again, I think we cannot even, there's not even for those slick cattle, there's not even an association by proxy, because the slick cattle are present in South American breeds. even though they're not necessarily in Angus breeds, we would have to genotype the beef for being Angus and then genotype it for being slick to say, okay, it's more likely that they've been generated through genome editing than natural. And also, what is the risk? I think that's where we really have to ask ourselves, what is the risk really? There's no risk because people have been eating slick cattle for generations. We have the data. Nobody's died from eating a slick cow. There's probably also, it's unlikely that there's even a taste difference, but we can test that, of course. So are we worried? And I think the similar question actually goes towards off-target effects, because we've recently seen a very good discussion about the natural variants, and Genus PIC, with whom we are actually running the PRRS resistant pig, have looked at 7,000 genomes, full genomes of natural offspring, and they found dozens of mutations per generation. So again, if there's one additional mutation that is maybe an off-target effect, and of course we can compensate for off-target effects or mitigate off-targets through in silico planning, do we care about another off-target effect, or is it just part of variation? If the off-target effect is damaging, then it will come out in the wash and these animals will never hit the market, whether it's welfare or general health or production traits. But if it doesn't impact production traits, just like natural breeding, the animal will just have more variance, but it's pretty much unrelated to whether this was generated through genome editing on target or off target even.
That's as comprehensive answer as I could dream of. What we've got to do is to, I think, identify off-target or on-target effects in 3 different categories. One is impact on or for humans. One is impact on the animals themselves, their well-being. And one can look at the Belgian blue and say, can we improve their lives and stop mastitis and so on? And the third is the impact on the environment. And I'm not sure that that one's been looked at as much as it ought to be, because at the moment we're talking about large animals. But we were talking about chickens earlier, and the escape of gene-altered salmon or tilapia may be very significant on the general population of these particular organisms in the environment. And you've identified that one as a major issue. And certainly we did over a long period of time. So can we assure ourselves that the impact on the environment is benign or good? It could be good because of the effects, as we said, of methane reduction. Can we identify the impact on the animal's welfare? And I think the answer is that we have to ask that question on every occasion we modify an animal. And the last one is, if we're doing it for humans, as we did earlier in this afternoon, is that something that we want to do, given the objection to modifying human embryos, particularly in the United States? So we've got to think about all of these issues when we're agreeing that the animal could be modified for livestock modification. We've also got a major issue, possibly, in terms of laboratory experiments, rather than livestock experiments and production of livestock, of laboratory animals, which may be very useful. But again, we have to ask all these questions in relation to those animals. I hadn't expected to spend so much time talking. I thought I was going to be simply moderating, but Francois, you're there. What should we do now?
Okay, so do you hear me?
Yes.
Okay, so sorry for this technical issue. One of the speakers is participating in another meeting. So he said that he didn't receive the Zoom link. So we are very unfortunate for that, but still, perhaps there's some other questions. I have one regarding the economical issue. So if I may, I would like to ask to the colleagues who kindly and nicely developed their technology and show how good it will be for food and for food dissemination, for food improvement. What's the economic model that is foreseen? Because if you compare to the drug designed, produced by genetic modification, there are very few on the market in low middle income countries. So I have the bad feeling that it would be the same with all this food developed by genome editing. Is it a good fear, or is the model we change because food is a priority as compared to drugs? Sorry for this naive question.
Anybody in the group who's willing to try and respond to Francois?
I'm not sure if I quite understood the question.
Question is if this food will be made available for low income countries. And I take the example of in parallel of drug that have been produced with genetic engineering, like monoclonal antibodies or hormones that are very, very, very few on the market in the South.
Yeah.
So that's a worry I have. But as I said, perhaps it's a naive question, and that all these have been anticipated by FAO or other international institutions. I don't know.
So I think if it comes to, go ahead.
Could I say something? Yeah, I was saying there is a big problem with this because of the genetic engineering technology is expensive to license. You know, it is a tool and it will be owned by someone. So if it's expensive to license, it will be very difficult to use in low middle income countries. So it will only be higher income countries who are developing this technology. So I think that's a big problem going forward. So we have to make sure that it's accessible to all to be able to use, and it isn't just involved in certain companies, you know, in higher income countries who are able to develop these tools and these plants and animals.
That's partially true, Mike. Very few of these technologies have been patented in Africa, for example.
Mm-hmm.
There's a small percentage of the technologies have been patented in South Africa, but for the rest of the African countries, there's very, very few. And I speak as an IP person. That means we can do it without asking the patent holder's permission.
Oh.
That creates problems as well. But in general, as long as it's done in one of the African countries rather than in a European country or the United States, and is only sold in those African countries, there is no problem.
Oh.
They can't come in now, 10 years after doing that patent, and say, we want to patent it now. That doesn't work. You've got to do it in the first year of the patent, okay? So there's been many questions about licensing, but as long as we check, and we have checked, I had students at the University of KwaZulu-Natal who checked where and what patents were being done in Africa as a whole, both in French-speaking and in English-speaking countries, and we found that there are relatively few. Okay, so it's better than we thought.
Good, thank you. I didn't know that.
Can I add something to this? Because here in South America, there are also many initiatives for the genome editing in animals. Well, you mentioned the one from Brazil, but in Argentina, there are also many models already not regulated, but that had undergone the regulation pathway. And I think it's the same situation as Julian said. It's case by case, and in some cases you can apply and you're free from to pay the license, and it depends on the application. If you're going to make money with that, maybe you have to pay the license, but if not, well, I think they evaluate that, and maybe in the future it will be more easier to get more models. But I saw we have a meeting last week with regulators from Brazil, Argentina, Uruguay, and Chile, Paraguay, and there are many models now arising, not only in plants but in animals and bacteria, microorganisms. And it's amazing after the CRISPR era how many new models we have in our region, and it's great for us because that means science is for everybody, and democratization of this great technology, and we can apply. We are a very small country here in Uruguay, but we already have developed this myostatin sheet model, and that helped a lot to develop us.
The nice one was for herbicide tolerance in Argentina, where then Monsanto failed to apply for the patent. So it was quite complex. Joaquin, you've asked a question. One of the main limitations in the use of this technology is that for politicians and the public in general, there is no difference between a transgenic animal versus an edited animal. Can we help clarify this situation where particularly politicians, I think you're suggesting, don't understand that a genetically modified and a genetically edited animal are different? Anybody want to come in and talk about that?
The reality is they're not necessarily different. They're different types of technologies, but the outcome is what is a question. I think we have to stop boxing technology so much, but actually think about what the eventual edit is. I think it's exactly what we've been discussing this afternoon. Is it cisgenic? Is it transgenic? Is it a knockout? Is it a small edit? And I think John tried to suggest one solution, which is what you do when you actually do you only regulate it when you bring in synthesized DNA at any step? Where do we draw the line? I think we have to draw the line somewhere, whether that is an arbitrary line or not. We have to make rules of sort or description. I think that is what we have to decide on. And I think that's where politicians are not wrong. They're reading genome editing, genetic modification, genome engineering. The reality is linguistically, they're relatively similar and encompass part of each other. So we cannot differentiate like that. We have to make rules how we actually regulate consequences of any of those technologies. And probably in the end, the technology itself is less important at the end of the day. So it comes back to that question of trait and technology regulation, where we again actually have different approaches between North America and South America as well, because whereas in South America, the actual outcome animal is basically regulated, whereas almost in North America, it's almost the technology and the type of edit can be regulated, but not each individual animal that comes out of that. So marginal differences, but huge outcome differences in how many animals can be produced on one license, etc. I think Angelica had a comment.
You're raising a major issue. Sorry, I haven't seen that. And that is, what's the trigger? We can't regulate every single new plant variety or animal type. We don't use the word variety in animals. Okay. we can't regulate every single one. So what will be the trigger that says this one's regulated and this one is not? When we wrote the Cartagena Protocol, and before in 1990, when we wrote the first regulatory structure for the EU, we said the trigger is genetic modification. Once you've triggered it, you then ask the question, what's the trait? But we were absolutely aware, and going back, I was involved in 1986, 87, 88. We can't look at every single new variety. So we have to have a trigger. And that's where the problem is. We can't say, it's just the trait we want to look at, because that's not going to work. So that's why we triggered it on genetic modification by the insertion or modification which could not have happened naturally. That is what we said in that piece of legislation. It's been misinterpreted over the last 30 years, but that's what we said. Okay.
But then there could be a next step. If the
Yes.
genetic modification is equivalent to a natural mutation, it goes in one direction, it can be approved.
Yes.
If not, it has to be evaluated.
That's exactly what we said at the time. As I say, those of us who wrote the legislation were aware of this, but a lot of people have misinterpreted what it says. Go back to the legislation and read it. It's really important. I think, for example, a deletion is not a genetic modification. That could happen naturally. Full stop. At least we thought so.
But I think that's exactly where people then are, well, what is, insertions can also happen naturally, but how big is that, can that insertion be? Does it have to be random? Can it be guided by an HDR, which is a synthetic piece of DNA? I think there's so many questions coming after that. And I think also coming back to your highlighting, we should look at environmental impact, off-target impact, etc. The problem is we cannot, it's almost like we're trying to regulate intense farming through regulating genome editing here. I think we have to separate those, because intense farming or farming in general must be regulated separate from genome editing, because genome editing is not the problem here. Breeding and intense farming impacts the environment, but that also already happens naturally. It's like the question around the PRRS pigs potentially allowing for denser stocking. It cannot be regulated within that. If disease would regulate our stocking densities, then we are really having a problem with our animal welfare regulations to begin with, because we could, well, of course we have in the past dosed animals up with antibiotics to allow for more intense farming. Then we brought in stocking density regulation. Now we're bringing in genome editing into this, but we still have stocking density regulation. So it's kind of that trying to regulate something within a law that is within regulation of something that's not actually linked to it directly.
I agree entirely. And actually, we did address that in the EGE opinion, so it's worth having another look. But yes, we were very aware that intensive agriculture, both with animals and with plants, obviously has a significant effect on the natural environment. And changes to field sizes is another example. Loss of natural habitat because of loss of hedges and so on is very significant in the UK, in France, and I guess those are the two I know. Whereas if you go to the Midwest of the United States, the farms are so large that it's less important. But we fail to understand in Europe. One of the things we talked about was in the United States, roughly 25% of the land which could be used for agriculture is. And in Europe, roughly 95% of the land that could be used for agriculture is. Okay? If you look at Namibia in Southern Africa, We can't grow crops, it's a desert, but we can grow cattle. So there's issues like that which need to be thought about. Have we exhausted this afternoon?
Can I ask about the pathway the regulation is taken now in Europe? Because I know most of South America, North America, but I know Europe was very restricted regarding that, and maybe that now they are starting opening their minds. Maybe Christine, Julian, Angelica, Mike can comment on that.
Anybody else? I don't know what the European Union is going to do about gene editing. They're still talking about it. The probability, in my view, is that it will be differentiated from modification. The European Court has decided that genetic modification and genome editing are the same thing, basically. I won't go into the breakdown of that. But the Commission is seriously considering whether a separation of those two should happen. We move to the member states because the regulation in Europe was essentially blocked from being applied by a relatively large number of member states saying we don't want this technology at all. And that was enough to basically stop the approval of many products which had been genetically modified. We have an opportunity with gene editing where we can differentiate gene editing from gene manipulation and produce new regulations for gene editing, not gene manipulation, which might result in more member states being accepting the technology. Again, I turn to the other panelists if they wish to comment on that.
But so you are in the process of discussing that, if this technology would be more easily accepted by the members.
Yes.
I think there's a mixture of trade pressure. As we heard today, we have animals that are genome edited that will enter the food chain in Europe, no matter what. There's also, I think, an economic pressure, especially with the drought that happened in Europe this year. There is pressure on actually growing crops that are more drought resistant, and the speed at which climate change is happening, traditional breeding cannot keep up with it. And I think the other thing that, again, I've been speaking to the European Parliament about this lately, and there is also the issue that we have that paradox where old technologies such as random mutagenesis are permitted, and the courts are, of course, in a bit of a pickle here because the only legislation that currently exists is about GMO. So regulation falls under GMO. So the bottom line is it lies with the European Parliament at the moment to put new legislation in place so that they could potentially be split. How quickly that will happen will depend on whether certain member states have objections to it, or how traditional breeds can be protected, how we're dealing with organic farming, all the things that are differently important to different countries. We have particularly France and Italy that are particularly precious about certain food items for good reason, but at the same time, they're also the ones that are probably the most affected by climate change. So there will be a middle ground that's both pushed by necessity, trade, as well as falling behind and protecting.
To help further, the only organization that can institute new legislation is the Commission. They propose legislation to the Parliament, and the Parliament can modify it, and then it has to be in agreement with the Commission and the member states through the Council. That the Commission is working on this quite hard, and there should be something coming forward fairly soon. But what that is, I don't know yet.
The result, but it's good news they are discussing about that.
They are discussing,
Right.
and
Yeah.
all member states were asked for comments. And the EGE was asked for comments, which is why I know about it. Although I'm no longer a member of EGE since the beginning of this year. I imagine they have been asked for further comment. I think I'm going to pass this to Francois and Hervé. I don't know where Hervé is, but we have reached the point where I think the end of the session is nigh.
Okay, so for the first time, we're very, very ahead of schedule, which is uncommon, but it's great for some of you have work to do. So thanks a lot to all the panelists and to you, Julian, for sharing this last session. I must say that when Hervé mentioned to you in the introduction, that before launching ARRIGE, we travel a lot around the world, and went in Latin America when we met Martina. And she was very lonely, I think, in Latin America for developing genome editing. And now, 4 years later, you mentioned that there are several experiments in different Latin American countries. So meaning that
it's amazing. Yes, how it grows here is amazing. Even in Paraguay or Chile, there is not countries that have been working before, and now they can manage to do that. So that's amazing, and we are grateful for that because we can develop further the science here.
The speed of development of this technology in human, in animals, everywhere is so fast that we thought important to have a look on the social implications of this technology, which is now under the radar, as we said. Nobody is talking about that, and we still need to have an ethical awareness of what is going on. So thanks to all of you. Just to say that ARRIGE is still alive after 4 years. And we have newsletter available on the website arrige.org, and you are welcome to join ARRIGE. And just to say that it is a second seminar. The first webinar was devoted to gene drive, which also it's another manipulation of animals with other issues. And we're preparing a next newsletter, and surely the annual ARRIGE meeting that will be held in Madrid, because our president is from Spain, and with Montpellier. Who is very sorry not to be here today. So thanks to all of you, and see you next time. Bye-bye.
Okay, thank you, everybody.
Bye-bye.
Bye-bye.